Learning with the Illuminati. Part 9. Introduction to the eye and vision. The eye. Some divisional parts of the eye are the cornea, the aqueous humor, the iris, the lens, the suspensory ligaments, and the ciliary muscles, the sclera, the choroid, the retina, and the fovea, and finally the optic nerve. The lens focuses the light on the retina. It is called myopia when the corneal surface is too curved, or when the eyeball is too long, and it is called hyperopia when the eyeball is too short, or the refracting system is too weak. The retina. From the eye cavity outwards, the retina contains the nerve fiber layer, the ganglion cell layer, the inner plexiform layer, the inner nuclear layer, the outer plexiform layer, the outer nuclear layer, the photoreceptor outer segments, and the pigment epithelium. There are 5 basic classes of neurons in the retina. These are photoreceptors, bipollar cells, ganglion cells, horizontal cells, and amacrine cells. Furthermore, the retina contains 2 types of photoreceptors, the rods and the cones. The shortest pathway of information from the photoreceptors to the optic nerve is via photoreceptor cell, to bipollar cell, to ganglion cell. The 2 other types of neurons in the retina are horizontal and amacrine cells. Both of these have their cell bodies in the inner nuclear layer. However, horizontal cells have their processes in the outer plexiform layer, and amacrine cells have their processes in the inner plexiform layer, which enables lateral interactions between photoreceptors and bipollar cells. Illumination. The receptors in the retina are in the dark, in a depolarized state, with a membrane potential of roughly -40 mV. Both sodium ions and calcium ions flows into the outer segment through membrane channels that are gated by the nucleotide cyclic guanosine monophosphate, abbreviated CGMP. The reduction in CGMP levels begins when a photon is absorbed by the photopigment in the receptor discs. These contains the light-absorbing chromophore, retinal, which is an aldehyde of vitamin A, coupled to one of several possible proteins called opsins. The different opsins tune the molecule's absorption to a specific wavelength of the light spectrum, allows functional specialization in rods and cones. In rods, in which the photopigment is rhodopsin, photons break one of the double bonds between the carbon atoms, and its configuration changes from the 11-cis isomer to all transretinal. The changes in opsin causes activation of an intracellular messenger called transducin, which activates a phosphodiesterase that hydrolyzes CGMP. Of great importance is that this biochemical cascade provides an enormous signal amplification. One light-activated rhodopsin molecule are estimated to activate up to 800 transducin molecules. Although each transducin molecule activates only one phosphodiesterase molecule. Further, each phosphodiesterase can catalyze the breakdown of up to 6 CGMP molecules. This results in that a single photon closes approximately 200 ion channels, or circa 2% of the amount of channels open in the dark, in a signal rod cell, which also causes a membrane potential net change of about 1 mV. In order to stop the cascade, activated rhodopsin is rapidly phosphorylated by rhodopsin kinase, enabling the aristin to bind to rhodopsin. which inhibits the activation of transducin, and the molecules are restored by the so-called retinoid cycle. Light adaptation. The photoreceptors are the most sensitive to light at low levels of illumination due to mechanisms regulated by the ion concentrations, but also due to neural interactions between horizontal cells and photoreceptor terminals. Due to having low spatial resolution, but high sensitivity to light, the rod system is specialized for sensitivity rather than resolution. Whereas the cone, which works different, has high spatial resolution, with low sensitivity to light, and allow animals to see color. As a consequence, a lot more photons are required to establish a perceptible signal from cones than from rods. At very low illumination, humans experience scotopic vision, whereas humans experience photopic vision during higher illumination. In intermediate conditions, humans experience mesopic vision. The retinal circuits do also contribute to scotopic and photopic vision. The signals from rods and cones may both go via the same ganglion cells to the nerve, depending on the level of illumination. And several rods may be coupled to a single ganglion cell, converging the weak signals to a stronger, however, reducing the spatial resolution. The total amount of rods in the human retina is about 90 million, whereas the amount of cones is approximately 4.5 million. However, the cone density increases almost 200-fold at the center of the fovea. Due to this overrepresentation of cones in the fovea, the light-sensitive rods are more activated in our peripheral vision, which explains why it is easier to see objects in the darkness by not staring directly at it. The cones can be divided into 3 different types, differing in the photopigment they contain, and are sensitive to different wavelengths of light, usually within the spectrum of blue, green, and red. Normal human vision is referred to as trichromatic. Deficiency in one of these causes dichromacy, and the most common color blindness are called protanopia, causing impairment in the perception of the longer wavelengths. Deuteranopia is caused by impairment of the medium wavelengths. Both of these have difficulties to distinguish between the colors green and red, thus the name green-red color blindness. Although very rare, it is called Tritanopia during impairment of short wavelengths, which is called blue-yellow color blindness.